And we're going to continue on looking at glucose oxidation and talking about cellular respiration and the 4 processes that are involved, involved in cellular respiration. And we left off on this slide, and we were sort of making the point here that when we break down glucose, we're talking about 1 molecule of glucose. And I just want to step back a little bit and remind us that glucose is only one example of a substrate that can undergo cellular respiration or metabolism. We can metabolize fats in a time of need. We can metabolize proteins. And so we're only looking at 1 molecule of glucose in terms of the context to explain these processes. But bear in mind that other substrates can undergo these metabolic processes as well, okay? All right, so going back to this slide, we said that if we break down 1 molecule of glucose, and that's the big picture that we're looking at today. We're talking about the steps involved in breaking down 1 molecule of glucose. And so what we will yield is some energy which can be used to phosphorylate ATP. I want you to keep that in the back of your mind as we go through the processes today. This is the end goal that we're trying to get to. We're trying to use the energy that's harnessed from breaking down glucose, not all of it, because we said this is not a 100% efficient process, but some of it is used to phosphorylate ATP as an energy store, as an energy reserve, which can later be used to do cellular work, okay? All right, and this is the main reason that we eat the food that we eat, right? To break it down, to get that energy, harness it to do some type of cellular work. Let's look at this equation again. Now, if we break down 1 molecule of glucose, we eventually yield 38 ATP. And theoretically, that should be about 686 kilocalories of energy. Okay, I'll say that again. Breaking down 1 molecule of glucose yields us 38 ATP, which theoretically should be 686 kilocalories. Now, in actuality, that's not the case. The amount of energy that's actually released is only 266 kilocalories. And so all of the energy that's actually liberated by breaking down glucose, we mentioned, is not transferred to ATP. Some of it is lost as heat. But look at how much, right? Most of it is lost as heat, 61%. We only get about 266 kilocalories from that 1 glucose when we know that there should be 686 kilocalories there. Okay, so the point of this slide is really to illustrate that this is not 100% an efficient process, okay? Most of this is lost as heat, and this speaks to us as beings on a bigger scale. We're only about 38% efficient in breaking down and harnessing the energy from the food that we're actually absorbing and eating, okay? All right, so these are the steps that are involved in glucose oxidation, and don't get too bogged down in the details that we go through here, especially glycolysis and the Krebs cycle. They have a lot of little steps, lots of enzymes. If you're going to med school, you're gonna have to memorize those, or graduate school. But for the scope of this class, we wanna keep with the big picture. We wanna understand where the energy is coming from and the big picture of the process. So these are the 3 processes that we're gonna look at. Glycolysis being the first one. And this is substrate level phosphorylation. So remember we talked about 2 mechanisms to phosphorylate ADP. One was substrate level phosphorylation. Can anyone remember the analogy that I gave for substrate level phosphorylation? I gave a little analogy to sort of distinguish these 2 mechanisms of phosphorylating ATP. So I talked about this as being analogous to stealing a phosphate as opposed to finding a free phosphate. So substrate level phosphorylation, we're taking the phosphate from a molecule, in this example, molecule X, and we're putting it onto ADP. This is by no means as efficient a process as oxidative phosphorylation. So that's what we're going to look at later on today. Oxidative phosphorylation, we find a free-floating phosphate and we use it to phosphorylate ADP. A much more efficient process, yields much more ATP, and we'll see that by the end of today, okay? All right, so let's start by looking at the steps of glycolysis. So basically, during glycolysis, we take a molecule of glucose, we bring it into the cell, and we start breaking it down. We start metabolizing it. And at the end of the 10 steps, there are 10 enzymatic steps involved in glycolysis. At the end of those 10 steps, what we have is a molecule of pyruvate. Pyruvate is the end result of glycolysis, and we actually yield 2 pyruvate for every glucose molecule that we break down. We also yield a couple of other things that are very important in this metabolic process. We yield 2 ATP, not a lot, right? We yield 2 ATP net, and I'll talk about why that's net in a little bit. And we also yield NADH+. So we reduce 2 molecules of NAD to NADH+. Does anyone remember what a reduction reaction entails? Or what happens with a reduction reaction as opposed to an oxidation reaction? So if you remember the mnemonic OIL, OIL, oxidation is loss and reduction is gain, right? To reduce NAD+ is to actually gain electrons. So basically this is nothing more than NAD+ taking up 2 electrons. And we'll talk about why that becomes very important later on. But the end products we yield are 2 ATP, 2 pyruvate, and 2 molecules of NADH+, and I want you to bear in mind that all that means it is carrying some electrons. And that's gonna head to oxidative phosphorylation, which we'll talk about in a little bit. So this is the important steps of glycolysis. Now there's no oxygen required here, so that makes this an anaerobic process, right? No oxygen required. And this occurs in the cytosol of the cell. Okay, the other steps we'll look at, or the final step we'll look at, takes place in the mitochondria. This is taking place in the cytosol. So let's look at the bottom of the slide here and look at the sort of reaction that represents the process of glycolysis. We put in our glucose, we put in 2 oxidized forms of NAD, and just to remind us, what is NAD? What biomolecule is NAD really? It's a nucleotide, right? Nicotinamide adenosine diphosphate. So this is a nucleotide, and basically this is in its oxidized form. We also invest 2 ADP, and then we invest 2 phosphates, and we get 2 pyruvate, 2 NADH+, and 2 ATP. Any questions on that? So this is the steps of the process that happens, and again, you don't have to memorize these enzymes or these steps, but what I do want you to understand is that for every molecule of glucose, when we get to G3P here, this splits off and happens twice. That's how we get 2 pyruvate for 1 glucose, right? So I'll say that again. So we're breaking down glucose, either the first energy investing stage of the reaction. When we get to G3P, which is this substrate here, this happens twice. So at the end of this, we end up with 2 pyruvate for every single molecule of glucose. And so as I said, we invested 2 ATP here, and down at the bottom, we end up yielding 4 ATP, right? Because remember, it's 2 here and 2 here, and this is happening twice. But because we invested 2 ATP, we're really only yielding a net of 2 ATP. All right, I'll say that again. We invest 2 ATP and we made 4 ATPs, 2 on either side of this leg here. And because we invested 2, the net of what we make is 2 ATP because we had to invest 2. Okay, I hope that makes sense. So we yield 4 ATP, which is really 2 net ATP. Okay, and again, this is another image sort of schematically showing the molecular changes of these substrates as they go through this process. I'll point out here again, when we get to G3P, this takes place twice, and so we yield twice the amount of products for each molecule of glucose. Okay, so any questions on glycolysis? Basically, the big picture, we're breaking down glucose, we're yielding pyruvate, we're yielding a little bit of ATP, not a lot, and then we're yielding some NADH+, which is simply shuttling some electrons to the mitochondria. Okay. Let's look at this linking step. So this is the second stage or the second step in the process of cellular respiration. So from glycolysis, our end product, which if you remember was pyruvate then enters this linking step, right? And all that happens here is this molecule of pyruvate under the influence of this enzyme, pyruvate dehydrogenase, becomes acetyl-CoA. We also yield some more electrons, right? We also reduce NAD+ to NADH+, which again is just simply picking up some electrons, and that's going to head to the mitochondria for oxidative phosphorylation. So along the way, right, through glycolysis, through this linking step, even through the Krebs cycle, we're generating electrons that are going to be going to the mitochondria, and we'll talk about them. So just bear them in mind as we talk about these. All right, so at the end of this linking step, our end product is acetyl-CoA, and bear in mind, for every glucose, this happens twice because we generated 2 pyruvate molecules. So pyruvate enters this linking step, we end up with acetyl-CoA, and we also end up with some carbon dioxide. This is the carbon dioxide that we breathe out. This is why it's very important for us to expire as human beings, not expire as in die, but expirate or let that carbon dioxide out as human beings, because these are the byproducts of these metabolic processes, and we have to get rid of them. Okay, this is how the body gets rid of them. All right, moving on to the Krebs cycle. So this is the third step or the third stage in cellular respiration. And again, this is another sequence of enzymatic reactions that you don't need to be too bogged down with, you know, in terms of the enzymes and the specific steps, but understand the big picture here. This is a cyclic metabolic pathway. This is the first process that takes place in the mitochondria itself. So the previous 2 processes we talked about took place in the cytosol, right, in the cytoplasm. This is the first step that actually takes place in the mitochondria. And at the end of this cyclical enzymatic sequence of events, we yield 3 NADH+, right, which again is simply 3 NAD+, grabbing some electrons and shuttling them off to the mitochondria. We also yield FADH2. Now, FAD is another carrier molecule, very similar to NAD. It carries less electrons and it's less efficient in terms of shuttling those electrons to the electron transport chain, but it also helps with that process. So we're going to count them as well. So 3 NADH+, 1 FADH2, we breathe out some carbon dioxide as a byproduct of this process, and we yield 1 ATP. So can anyone tell me how many ATP we yielded so far? So through glycolysis, we got 2 net, right? How many did we get in that linking step? Let's jump back. We didn't get any here. How many did we get in this step? We got 1, right? But because we're doing this twice, how many did we actually get? 2, right? So far we have 4 net ATP, okay? All right, and let's just remind ourselves of the overall reaction down here. We're investing acetyl-CoA, which is the substrate that's coming from that linking step. We're investing NAD+, FAD, some ADP, and we're going to phosphorylate that at the end to make ATP. We're going to reduce our carriers here, so NAD and FAD, we reduce them by adding some electrons, and we're also yielding some CO2, which we breathe off. Okay. All right, and this is the exact, or the sequence of events that is included in the Krebs cycle. The Krebs cycle is also called a TCA cycle or the citric acid cycle. Those are some synonymous terms. And again, don't get too worried about the steps here, just the important substrates and the important end products that we yield. All right, any questions on those 3 steps? Okay. Mm-hmm.
On the previous slide, so are you only generating 1 ATP or how many? Yeah, sorry.
So you're generating 1 ATP for each acetyl-CoA, okay. But remember we had 2 pyruvates, so we have 2 acetyl-CoA, so we're really generating 2 ATP. Mm-hmm. Any other questions? Okay, so we're gonna get a little bit more complicated now. We're gonna talk about oxidative phosphorylation, and this is the final stage or the final step in cellular respiration. And this is really the most important. This is the step that requires oxygen. We're going to talk about the unique function of oxygen in the electron transport chain. And this is also the step that occurs across the inner mitochondrial membrane. Also, we want to bear in mind that this is a much more efficient way of making ATP, a much more efficient mechanism than substrate level phosphorylation, right? Oxidative phosphorylation is going to yield us much more ATP than the 4 ATP we talked about previously, or up until now. Okay, so most of the ATP is formed here, and it's basically formed by electrons moving down the electron transport chain. So one electron, those electrons that we've been talking about, that I told you to sort of keep in the back of your mind, right? Those carrier molecules, FAD and NAD, remember, they're carrying electrons from these processes in the cytoplasm into the mitochondria. These electrons are then going to be passed through a series of complexes. And as they go through these complexes, they're eventually going to create what we call this proton gradient, okay? This process requires oxygen. That's very important, hence the term oxidative